Welcome to the session on nutrition today. I'm the new president of the Commission for Bee Biology. My name is Geraldine Wright, and I'm in the Department of Zoology at the University of Oxford. It's a great pleasure to be here and to see all of you and to see friends that I haven't seen in a long time. And it's a lot of fun to be the chairperson for this session on nutrition. Hello? Okay. So, this speaker that was supposed to speak in this slot didn't upload their talk, and we're not sure if that person is here. So you might be getting a mystery talk from me. Now, I've given them a talk that's on a USB that I gave 1.5 years ago, and I haven't looked at it since. So it really will be a mystery talk, but it will be on bee nutrition. If the speaker that's supposed to speak right now doesn't come here in 10 seconds, I'm going to move on. I need to time myself or we'll be here for a while. Okay. Go. Bees, as you know, they live in large colonies that make up thousands and thousands of members. We have members within the colony which eat the pollen and the nectar, and those are the nurse bees, the younger bees, and we know this from Professor Krailsheim's work. And we have the foragers, and the foragers don't really eat the pollen. They eat nectar and carbohydrates and honey, and they get fed glandular secretions by the workers. And pollen is really very important for honeybees. Pollen is their source of protein and fat. It also contains important micronutrients like minerals and vitamins, and all kinds of other phytochemicals, like Amy said. But really, it's their main source of protein and their main source of fat. And we know that most floral pollen, actually this is some data that was collected in my lab and then never published, unfortunately. We know that pollen is, at least from the study that we did of UK pollen, is about between 5 and 10% protein, which actually, in this study, it seems a little bit low compared to the literature. The literature, its average is around 30% protein, but it's, the point is that protein is highly variable in pollen. And what I took away from Christina Grosinger's talk yesterday is that of the macronutrient components of pollen, that protein actually is less variable than fat. That's a very interesting fact that I didn't know. This is a really nice study that was done by Dong et al. And published in the Journal of Apicultural Research. They report that fat in pollen ranges from about 3% to about 10% of the pollen. You can get extreme values like 20%, but on average, pollen is between 3 and 10% fat. And these are the major plant families that honeybees forage on in the landscape. So their main form of carbohydrates is floral nectar. Floral nectar is mostly sugar, as we all know. And bees have to balance their intake of these macronutrients. And what's interesting about honeybees is that the macronutrient nutrition is partitioned so that you have carbohydrates basically coming from floral nectar and floral nectar, and your protein and fat coming from pollen. This was a little bit like our diet, actually, interestingly. We can get our carbohydrates from things like sugary drinks, and we can eat our protein and fats from the meat that we eat. Ooh, that advanced without me even touching it. So, and what I want to do is to explain to you the rationale for the study that we did to try to understand how bees regulate their intake of protein and fat. So that's what this little talk is about. It's about some work that was done in my lab to look at how nurse bees regulate their intake of protein and fat when you give them a chemically defined diet in the lab. So the geometric framework for nutrition is a modeling framework that was developed by my PhD supervisors at Oxford, Steve Simpson and David Robenheimer. And its premise is that all animals have to optimize their intake of macronutrients simultaneously. So every food you eat is a combination of protein, carbohydrate, and fat, and you and your physiology have to optimize your intake of those macronutrients to meet your nutritional needs. So we have physiological mechanisms that have been shaped by natural selection to permit us to do this. And the point of this slide is really to show you that, I just need to remember what was there before, okay. I told you I didn't see this, so I'm winging it. This is in our diet is a mainly carbohydrate source. This one's mainly protein, but some fat. And then this is the happy medium, right? So this is the perfect diet for people. It has its carbohydrates on top and the protein and fat in the middle. So the principles of nutrient balancing are that you can, in a chemically defined diet in the lab, you can look at how animals move through this nutritional space if you give them a chemically defined diet that has different ratios of these macronutrients. So, and this is a graphic example of this. So this is the protein axis and this is the carbohydrate axis, and this would be your steak rail. Okay, so it's high in protein, so it's way down here and not too much carbs. And here's your pasta, it goes up here, it's not too much protein. And in the middle is our intake target. That's what it's called, the perfect ratio of nutrients that we need. And you would eat in any given series of meals. Someone tracked you through time. You would eat a series of meals that consisted of pasta in one meal, protein in the next, pasta, protein, until you get to this happy medium in the middle. So that's the premise of this modeling, is that by watching what an animal does, if you give them choices of food, they will tell you what their optimum macronutrient intake is. So with bees, it's very interesting again that their food is partitioned. You have it partitioned into carbohydrates on this axis and protein and fat on this axis. And our point was really to try to use chemically defined diets to find out what bees actually need. I need to hurry up because I'm going to run out of time here. And so we use diets to study this. With honeybees, again, I worked with, in our diets were composed of essential amino acids, fat, and carbohydrate, or just sugar. So we gave them the option, if they wanted to just eat sugar, they could, or they could eat a diet that was composed of essential amino acids, the 10 essentials, fat in the form of an emulsion of lecithin and some oil, and carbohydrates. And these varied in terms of the ratio of the essential amino acids to fat, so we could figure out what they wanted. Those are the rails. Okay, I'm gonna move on. Those were the specific proportions of the amino acids and fat in the diets that we tested. And in previous work that I did with Anthony Baudo, who was in Christina Grosinger's lab, we found that bumblebees, using this approach and these particular diets, we found that bumblebees regulate their intake to a diet that was about 14 parts amino acid to 1 part lecithin, which is actually pretty dilute in fat. It's pretty high, relatively high protein diet. And that's for a forager, not for a nurse. And they have a slightly different biology. So we use nurses. We were interested in the nurses because they're the ones that eat the protein. They're the ones that make the royal jelly that gets fed to all the larvae, and that's a hugely demanding task, requires a lot of protein, a lot of fat. So this was the design of this particular experiment. The most dilute in fat was 25 parts amino acids to 1 part fat, and the most high was 1 part amino acids to 5 parts fat, which is a pretty fat diet. And that's where pollen sits, is in about this range. And this is the technique we used. I'm running out of time. We used this box. These are bees in the box. We kept them in there for 2 weeks. They got to feed on any of these diets, and the diets were presented in these tubes, and we measured it every day. So we measured the amount of food, and we measured how many died. And what we found on average is that the total amount of food was more or less the same in the diets that we tested, and this is the diet that varied here on the axis. This is protein to fat, or amino acids to fat. But what's interesting is that the intake of the amount of this diet, the amino acids and fat, actually increased in the animals that were fed with the diets that were high in fat. So if there's more fat in the diet, the bees actually eat more food. Sorry, there you go. And it was very interesting then to look at how they regulate around a specific ratio of amino acids and fat in the food. On this axis is the amino acids, on this axis is the lipids. And this is the variable diet in our experiments. This again is the low-fat diet. This is the high-fat diet. These two are the highest fat diet, and they're closer to what's actually in pollen. And what you can see here is that the distance that you take along this axis means that's more food, more of this reagent eaten. This is more of this reagent eaten. And the animals that were fed with the high-fat diets eat more food in total. And they also regulate their intake to a proportion that's about 1 part amino acid solution to 3.3 parts fat. The ones that were fed on the more dilute fat diets actually regulate to an intake of about 1 part amino acid to 5 parts fat. So it's quite interesting. They're not radically different, but the ratio that they go for is about is between these two. But what's very interesting here is that when they're fed on a high fat diet, they eat more food, like us. Like me. So again, this is just the same diets again. This is the low-fat diet, this is the high-fat diet, this is the amount of amino acids, this is the amount of carbs. And interestingly, the ones that were fed with a high-fat diet, and this is the proportion of carbohydrates to fat, they eat proportionally more fat on the diet that has more fat, or in the diet pair that has more fat. And this is even though they're given ad libitum carbohydrates. The ones that are fed with the more dilute fat diets and the choice of diets, they eat proportionally more carbohydrates. Okay, and this is just a comparison again from this experiment of the amount of lipids and carbs. Oops, sorry. Forgive me again, I haven't looked at these data in a while. This is carbohydrates to amino acids, this is carbohydrates to lipids. So more or less, they regulate around an intake of about the same lipid to carbohydrates here. It's about 1 part lipid to 33 parts carbohydrate. So in summary, we found that newly emerged honeybees regulate their amino acid to lipid ratio in our chemically defined diets around an intake target of approximately 1 part amino acid solution to 3 parts lipid. Now the amino acids in this solution, you could choose to put them in any ratio. We put them in at the same concentration throughout, just to standardize it. It's interesting, but protein isn't like that, and I have more data about that, but I'll present it at another time. Here, just to standardize it though, using these particularly chemically defined diets, we find that they regulate around a ratio of 1 part amino acids to 3 parts fat. That's a fairly high fat diet. The animals fed with a high fat diet eat more food and they eat more amino acids. The intake of lipids to carbohydrate is regulated to about 1 part lipid to 35 parts carb. And this is lower than in bees that were fed with no fat. So if you feed them just with, sorry, I can't remember why I said that. I'm gonna move on. We can talk about it after. And what I'm interested in this, this particular study is to understand actually whether the fat that's in the diet is actually affecting the physiological state of the bees, and I think that our data indicate that. When the bees actually have more fat in the diet, it changes their physiology and they actually eat more, and it probably ramps them up into hypopharyngeal gland production, so they're producing more jelly. We have more data, but I can't show it just now. So in summary, we think that fat in diet is really important for bees, as actually as Amy Toth has shown in her previous work, and that it could be that the amount of fat in the bees' diet is actually probably having a pretty strong effect on their physiology. And this work was done in collaboration with Sharoni Shafir and Phil Stevenson, who've been my partners in crime for many years. Just going to check my time. I think I'm even ahead of time. My postdocs, Dan Stabler, Mushtaq El-Asawi, and Alex Robinson, who was one of my students, were the people that actually collected the data for this study. And my beekeeper at Newcastle University, where I used to be before Oxford, was Malcolm Thompson. And Malcolm helped me for years. He was a retired fellow who always helped me and was my great friend. So all the bees that were used in this study were from Malcolm. And this was funded by the British Biotechnology Research Council, the BBSRC. And I'll take questions at the end of the session. So if the next speaker is ready to come to the front, please come to the front, and thank you very much.